In this video, we're going to learn all about ATP synthase. Now, ATP synthase is made out of two basic parts called the F1 part and the F0 part. F0 is in the cell membrane, that's what's pumping your hydrogens. F1 is a membrane protein, water soluble in the cytoplasm. That's going to be making our ATP, and they're connected by a stalk. You notice here that here we have some blobs. That means we don't know what the structure looks like, but for the most part, we have the structure of ATPase solved, and it looks like just from looking at this, we have a ton of alpha helixes with a little bit of beta sheet right there. Now, the F1 component, that's our water-soluble component, is made out three alpha subunits, three beta subunits, a gamma, a delta, and an epsilon subunit. And it's a big protein that's connected to a 50 angstroms long stem that's here in the blue. Now our alpha and beta subunits, so if we take our molecule and look at it looking down the stem, you can see that we have three alpha subunits and three beta subunits. And you can think of these as being linked together. So alpha beta pair here, alpha beta pair here, alpha beta pair here. So we have three pairs of alpha and beta, and these are 20% identical to each other. And they are alternating, like I said, and you can also think of them like an orange or a fruit that is naturally segmented. So the lower portion of our helix down here is forming an anti-parallel coil, so remember that from Biochem 1, with the N-terminal segment of our gamma subunit. This coiled delta and epsilon subunits is what's allowing F1 and F0 to be linked together. Now beta subunits, these are the things that are going to make ATP for us, and we have three major conformations that we care about. They're called DP, which is binding ADP, TP, which is binding ATP, and E, which is empty. So finally, in biochemistry, we have letters that make sense to what we're actually doing. And that's mainly because ATP synthase has been discovered relatively recently. Now let's look at how ATP synthase works. So basically, ATP synthase has three main things it has to do. One, we have to move protons with the gradient. This is carried out by F0. Two, we have to make ATP. This is carried out by F1. Three, we have to match this translocation of protons to the formation of ATP. So we need the interaction of F1 and F0 to be coupled. Now we're going to look at our F1 segment first. And so we have three different AB units. And like I said, we have three different conformational states. L, T, and O. These are also called, like previously, DP, TP, and E. So L, T, and O will switch with each subunit. So here we're going to be getting the 1O, 1L, 1T. And during this, these alpha beta pairs, so an O is an alpha beta pair, L is alpha beta pair, T is alpha beta pair, these things aren't really moving. Rather, our protons in the F0 subunit, that energy is going to be used to just change the conformation of these subunits. So we begin with ATP in our tight conformation, and an ADP and phosphate are going to come in and bind our loose conformation. Protons are going to go through. Three protons are going to come through, and we're going to use this energy to switch our conformation. So a tight is going to, or a T is going to an O goes to an L goes to a T. So we're going counterclockwise here. And in the O state, we can't be bound to any products or substrates. So ATP is released. In the T state, we bind our products tightly. So the ADP and PI are going to be forced together to make ATP. The L state, our substrate and products bind loosely. So we're going to bind another ADP and start the cycle over. So that's what's going on in our F1. That's how we're making ATP. And you can notice that during this energy, this green thing actually changed conformation. That's our stalk, and that is how F0 and F1 are communicating with each other, and that is how our energy is being moved. The energy from the proton gradient, that is. So this basically just states everything I just went through and our different states. And here we have our dp and our tp and our E again. So feel free to read this over to basically just go over what I just said. And like I said, this gamma subunit is going to be our crank. So here we go. Here's the whole model again. Let's focus on F0 now. So F0 has this ring of C subunits. This is what's going to be moving, while our A3, B3, F1 unit, this isn't going to be moved. This is stationary. C's are moving. And this rotation of the C is what is allowing the protons to go from the periplasmic space to the cytoplasm space. And what's happening is that a proton will enter our hydrophilic channel between the C ring and our alpha subunit. We don't know what the alpha subunit looks like, but we know there's some kind of channel there that is allowing protons to bind to the C subunit. When a proton binds, the C subunit will start to move in our counterclockwise direction. It takes roughly a full turn before the proton can be ejected into the cytoplasm. So this proton will have to go around roughly 90% of the way until it's finally ejected. Every complete turn will generate 3 ATPs. Now, the C subunit, here's what the C subunit looks like. It's really just two helices that are different lengths. And you have a 4-substrate polar or 4-residue polar loop that's arranged in an anti-parallel coiled coil. Now, what's happening is that the proton probably binds to aspartic acid 61. And this protonation will change the conformation of this subunit. So that's going from A to B. So A, you can see we're a little bent. B, we're now straight because of our protonation. This change in conformation is causing a mechanical push against the subunit, which is giving us energy to rotate our ring. So every time we're getting a proton, we straighten out. That straightening out causes a push, and we rotate, and we keep adding hydrogen to new C subunits to rotate until you get 90% of the way through. Then you pop off your hydrogen because you need to accept a new one on our next movement. Now, how do we know this works the way I just said? Well, a very cool experiment took an actin filament and basically attached it to the C subunit, and it was labeled with fluorescence. So we could watch this. And what happens is that you'll see the actin actually move through time. And it kind of does so in a jerky motion. So here actin move. So it kind of just jerks. It's not like a fluid movement like you would actually expect. To kind of move, stops. And we're actually able to visually see, yes, in fact, the C motor does move in a way that has been predicted in this counterclockwise manner. Now, it is possible to uncouple ATP synthesis from the electron transport chain, and we talked about that. And one way is a molecule called DNP, or 2,4-dinitrophenol. What DNP does is that it will go to our intermembrane space, bind a hydrogen, and then it can diffuse into the matrix, and it will deposit that hydrogen. Basically, you break the proton gradient with this molecule. And DNP was an effective diet pill in the 1920s. So and it makes a lot of sense why it's a diet pill. So we go through glycolysis, we go through the citric acid cycle, all of this to make NADH, FADH2, and a few ATPs, but mainly the NADH and FADH2. We're going through the electron transport chain solely for the purpose of pumping hydrogens out against the gradient. This molecule is breaking that step, so the body is saying, well, I'm still low in energy, my ATP is low, let's start breaking down fat, let's start breaking down protein. We need more things to go through glycolysis, more things to go through the citric acid cycle, but if you have DNP, you're never going to get enough. So sure, you lose weight, but there were some side effects that were, fatal. So you can't really buy DNP anymore since it will probably kill you. But you do lose weight, so I guess it's a double-edged sword there. And with that, that's the end of this video. I will see you in the next one.